Hello guys, today we're going to talk about the sterilization and disinfection. Well, this is the very important topic in the medical field because we use them every day in our medical life and practice. So let's first start with the definition and the terms. Sterilization. It is the process that remove, kills or deactivate all form of life such as fungi, bacteria, virus along with their spore. So in sterilization there is a completely removal of all form of life and along with their spores, even their spores are destroyed. So after an sterilization, an object is referred to be sterile or aseptic. It is the 100% removal of all form of life. So this is the sterilization, and there is a completely removal of all form of life along with their spores. So these spore cannot be developed into the future pathogens. So this is the sterilization. Disinfection. It is the process of elimination or killing most pathogenic microorganisms, but not their spores, from an object or instrument. So in disinfection, there is all removal of all form of life, while the spore remain unaffected. So this in disinfection, it's like the 90% removal of all form of life, but the spore are not killed. So this spore could develop into the future pathogens. So disinfection, it's like the 90% removal of all form of life. And the chemicals that are used for disinfection are called disinfectant. So we can use different types of disinfectant such as Dettol, Lysol, alcohol. So this helps to kill some most of the pathogenic microorganism, but while their spore remain unaffected. And we have two types of disinfection. So one is the oxidizing disinfectant. So this kill the microorganism by oxidizing the contents of the microorganism. So some of them are sodium hypochlorite, peracetic acid, hydrogen peroxide. And the next one is the non-oxidizing disinfectant. So they kill the microorganism by they penetrate the cell wall and disrupt the phospholipid molecule, and this block the metabolic pathway of microorganism, which eventually leads to its death. So the example are quaternary ammonium compound and also bisbiguanide. Next one is decontamination. It is the process of removal of contaminating pathogenic microorganism from the article by a process of sterilization or disinfection. So in decontamination, it's like the removal of contaminating bacteria by either sterilization or disinfection. We can use both of them, and this uses physical methods such as heat or different type of machines, autoclave, hot air oven, or also we can use the chemical methods such as Lysol, sodium hypochlorite, hydrogen peroxide to remove the microorganism so it no longer remain the infectious. So that's the decontamination. It's like removing the contaminating bacteria from an article. So next one is the sanitization. This is the process of chemical or mechanical cleansing applicable in public health. It is usually used in food industry. It reduces microbes on eating utensils to a safe level for public health. So sanitization, it's not the completely sterilization, but it's like the cleaning the area at the safe level. So mostly used by food industries. The next one is asepsis. So this is the technique such as use of globes, air filter, UV rays to achieve microfree environment. So in this process we use different types of materials such as globes in the laboratories or air filters in these operation theaters, UV rays to kill the microorganism, and the antisepsis. This is the use of chemicals to make skin mucous membrane free of pathogenic microorganism. So in antisepsis, we use chemical to remove the microorganism, while in asepsis we use the materials and such as gloves, air filters to remove the microorganism. And the substance that are used for antisepsis are called antiseptic. So whatever the subchemicals that are used are known as antiseptic. These either kill or inhibit the growth of microorganisms from living tissue. So these chemicals, they either kill or inhibit the growth. Examples are chlorhexidine, povidone, iodine, alcohol. These are called antiseptic. Next one is bacteriostasis. So it is the condition where multiplication of bacteria are inhibited without killing them. So bacteriostasis, they only inhibit the growth of microorganisms, but they do not kill them. The next one is the bactericidal. It is the chemical that can kill or inactivate bacteria. It may be called bactericidal, virucidal, fungicidal, microcidal. So bactericidal, this is the chemical such as antibiotics or other kind of chemical that can kill the bacteria or inactivate them. So they are called bactericidal. If it kills the virus, then it is called virucidal, or if it is fungi, it is called fungicidal, or if it is other form of microorganism, it is called microcidal. So these are called bactericidal. Next one is the antibiotics. So they are the substance produced by one microorganism that inhibit or kill the growth of another microorganism. So the techniques of sterilization, we have different types of technique. The most commonly used are physical technique, chemical technique, and physiochemical. So in physical technique, we have sunlight, heat, and vibration and radiation. Also in heat, we use two type of heat. One is dry heat and other is moist heat. In dry heat, we have red heat, flaming, incernation, hot air oven, and infrared. While in moist heat, we use the heat below 100 degrees Celsius, at 100 degrees Celsius, above 100 degrees Celsius. So these are the types of heat. In radiation, we use two types of radiation such as non-ionizing radiation, ionizing radiation such as electromagnetic and particulate. So these are the physical types of sterilization. While in chemical, we use different types of chemicals such as in the liquid form we use alcohol, aldehyde, phenolic, halogen, heavy metals, surface active agent, dyes. Also we use the gaseous form of chemicals such as formaldehyde, ethylene oxide. And this physiochemical is the combination of both physical and chemical method of sterilization. So these are the techniques of sterilization. So let's check one by one. Now let's see the physical method of sterilization. The first one is the sunlight. The microbicidal activity of sunlight is mainly due to presence of ultraviolet rays in it. So it is responsible for a spontaneous disinfection in natural condition, and it acts as a natural disinfectant of water bodies like tanks, lakes, because of ultraviolet rays in of the sunlight. So we know sunlight, it has UV rays, and these UV rays are very effective in killing microorganisms. So they penetrate the microorganism cell wall and disrupt their membrane, so it helps to kill the microorganism. And this is the natural one, so this is the great source of disinfection in natural condition. The next one is heat. Heat is the most reliable method of sterilization. So it's of two types. One is dry heat and moist heat. And dry heat act by protein denaturation or oxidative damage. So dry heat kill the microorganism by denaturation of protein of the microorganism, or they oxidize the chemical present in the microorganism and kill it, while most heat kill microorganisms by coagulation and denaturation of protein. Moist heat is much better than dry heat. So moist heat kill microorganism by coagulation of protein. So microorganisms are, they have a protein in them, so it coagulates the protein, so it kills them. And we have the important term here, thermal death time. So this means it is the minimum temperature that is required to kill microorganism at specific temperature. So in the dry heat, we use different type of dry heat such as red heat. So in this type, articles such as inoculating loops, straight wires, tip of the forceps, spatula are sterilized by holding them into the Bunsen burner until they are red hot. So this is the simple method that provides sterilization. And the next type of dry heat is the flaming. So articles such as scalpels, mouth of the test tube, flask, glass slides, or cover slips are passed through the flame for few minutes. This provides some sort of disinfection. And the next one is the incernation. This is the method of destroying contaminated materials by burning them in the incinerator, such as dressings, animal carcasses, pathological materials. So next type of dry heat is the hot air oven. It was introduced by Louis Pasteur, and this is nowadays the most commonly used type of dry heat. And the materials to be sterilized are kept at 160 degrees Celsius for 1 hour, or it can be also kept at 170 degrees Celsius for 40 minutes, or also 180 degrees Celsius for 20 minutes. So these are the procedure for sterilization. And the instruments that are used for sterilization in hot air oven are instruments such as forceps, scalpels, scissors, glassware such as petri dish, pipette, flask, all glass syringes, swabs, oil, pharmaceutical products. So these are the instruments that are used in the hot air oven. And the next type of dry heat is the infrared. So in this type, materials such as metallic instrument and glasswares are sterilized by passing them through infrared tunnel. It is used for special big laboratories. So those were the dry heat. Now let's go for the moist heat. So moist heat kill microorganism by coagulation of protein at high temperature and pressure. As we know, bacteria or any microorganism, they have protein in their cell wall or cell membrane. So when they are exposed to the moist heat, this moist heat coagulates the protein and it kills the microorganism. So we use different types of moist heat. That we Let's see the moist heat below 100 degrees Celsius. So the moist heat below 100 degrees Celsius is used in pasteurization, and this is mostly used in dairy industries. Moist heat below 100 degrees Celsius are sufficient to kill the microorganism of milk, such as Salmonella, E. coli, or other microorganisms that are present in the milk. It is also used in vaccine bath. So the contaminating bacteria in a vaccine preparation can be inactivated by heating in the water bath at 60 degrees Celsius for 1 hour. So this is the procedure that are used in vaccine bath. Also it is used in serum bath. So the contaminating bacteria in serum preparation can be inactivated by heating in a water bath at 56 degrees Celsius for 1 hour. So moist heat below 100 degrees Celsius is also used in the serum bath, which kill the pathogenic bacteria. So let's see the temperature above 100 degrees Celsius. So moist heat above 100 degrees Celsius are mostly used in autoclave. So autoclave, it is the most widely used form of sterilization, and sterilization can be effectively achieved at temperature above 100 degrees Celsius using autoclave. So in autoclave, the pressure of 15 lbs and temperature of 120 degrees Celsius is used for 15 minutes to sterilize the instrument. So this is the principle of sterilization, at which the pressure of 15 lbs and the temperature of 121 degrees Celsius is used for 15 minutes. So the materials that are sterilized are culture media, dressing, certain equipments, gowns and clothes, and all theaters appliances. So these are the instruments that are used in autoclave for sterilization. And the next one is the radiation. This is the another type of physical technique of sterilization. So we have two types of radiation. One is the ionizing radiation and the other is non-ionizing radiation. So in ionizing radiation, gamma rays are used, and these gamma rays, they have great penetration power. So they are used to sterilize disposable dishes, plastic syringes, antibiotics, vitamins, glasswares and fabrices. So in ionizing radiation, we use this type of instrument, while in non-ionizing radiation, UV rays are used for sterilization, and they are used to disinfect hospital wards, operation theaters, virus lab corridors. Next one is the filtration. Although it doesn't kill microorganisms, but it separates them out, so this is also a little bit effective. The object or instrument which cannot be sterilized by autoclave or hot air oven process, filtration is used. And we have different type of filters such as earthenware filters, asbestos filter, sinters filter. Now let's see the chemical method of sterilization. So we have different type of chemical that can be used for sterilization, and they are either in the liquid form or in gaseous form. So let's see all the types of chemicals one by one. Alcohol. This is most available and widely used form of sterilization, and 70% of ethyl alcohol is used as antiseptic. So antiseptic, it is just a technique to remove the pathogenic microorganism, and it is also used to disinfect the thermometer, so it helps to remove the pathogenic bacteria from thermometer. And the next one is the aldehyde. The examples are formaldehyde, glutaraldehyde. And what are the uses? So 40% of formaldehyde is used for surface disinfection, fumigation of room, chambers, operation theaters, ward, sick room, while 2% of glutaraldehyde is used to sterilize thermometer, bronchoscope, and aesthetic equipments. The next types of chemical compound is phenol. The examples are phenol, cresol, lysol, chlorhexidine, and the uses of them are: phenol is used to disinfection of ward, floor, bed pans, while chlorhexidine is used for skin disinfection. Also, it is most widely used in mouthwash. Next one is the halogens. The examples are such as chloride, iodide, and the uses of them are: 2% of iodine with 70% of alcohol is antiseptic. So this remove the bacteria or not virus, but other form of microorganism. And the 10% of povidone iodine is used as skin disinfection in operation theaters. So this 10% of povidone iodine is widely used as disinfection in operation theater. Next one is the ethylene oxide. This is highly effective in killing microorganisms, including spores. So it is used for sterilization of bedding, textile, rubber, plastic syringes, disposable, petri dishes, dental equipments. So ethylene oxide, this is very effective, like it kill all form of life, including the spores. So there is 100% removal of all form of life. The last one is the physiochemical method of sterilization, in which both physical and chemical method is combined for the sterilization of instrument.